Hi friends, once again welcome back to my channel. Today we are discussing cells of immune system. Before that, we must know about the term hematopoiesis. Hematopoiesis, it is the formation of blood cells. Formation and development of blood cells, it is hematopoiesis, and it is starting in the embryonic yolk sac. That is at the first week of gestation itself, hematopoiesis it is starting in the embryonic yolk sac. And all the blood cells, they are arising from a single type of cell, that is a hematopoietic stem cell. It is the parent cell. From this hematopoietic stem cell, all the blood cells are arising. This is because hematopoietic stem cell, it is a multipotent self-renewing cell. So it can differentiate in different ways to form RBC, WBC, mast cells, dendritic cells, platelets, etc. These hematopoietic stem cells, they are adopting two pathways to form two progenitor cells. Here we can see a myeloid progenitor and lymphoid progenitor. That is, the two progenitors are hematopoietic stem cells. They are giving rise to two progenitor cells. They are myeloid progenitor and lymphoid progenitor. Then this myeloid progenitor, it gives rise to dendritic cells, RBC, basophils, eosinophils, platelets, monocytes, neutrophils, and these monocytes, they give rise to macrophages. From the myeloid progenitor we can see for each of these cells they have progenitors. That is, for RBC we can see an erythroid progenitor, a basophil progenitor for basophil, eosinophil progenitor, megakaryocyte progenitor for platelets, and granulocyte monocyte progenitor for monocytes and neutrophils. The lymphoid progenitor, that is the second progenitor, it gives rise to dendritic cells, natural killer cells, then T cell progenitor, B cell progenitor. And the T cell progenitor, it gives rise to T cells. That is, the main two subsets are T helper cells and T cytotoxic cells. B cell progenitor, they give rise to B cells. The most important type of cells in our immune system are lymphocytes. Based on the functions and the cell membrane components of lymphocytes, they are classified into B lymphocytes, T lymphocytes, and natural killer cells. Here, first one is B cells. These are mononucleate cells. They are non-granular cells. They are having a large nucleus and a small rim of cytoplasm. And the mature B cells, they have the surface bound or membrane bound immunoglobulin molecules. This is the membrane bound immunoglobulin molecule. And the B cells, these are the receptors. These are the typical B cell receptors. And the B cells, they are playing an important role in humoral immune response, that is antibody mediated response. Then receptors of the B cells. The main receptors of the B cells are listed below. They are B222, it's a marker for B cell, class 2 MHC, that is the presence of class 2 MHC molecules on the surface of B cell make them an antigen presenting cell. And the CR1 and CR2 receptors, these are the receptors for the complement factors. Then Fc gamma R2 receptor is present on the surface of B cell and is a receptor for immunoglobulin G. Then B7-1 and B7-2 receptors, and these are interacting with the regulatory molecules of T cells. CD40 receptor, it interact with the CD40 ligand which is present on the surface of T helper cells. Then coming to T cells, they derive the name from the site of maturation, that is thymus. The two main subpopulations of T cells are T helper cells and T cytotoxic cells. And the T helper cells, they are more present in our human blood or in our blood than cytotoxic cells. That is, the ratio is T helper TC ratio is 2:1. TC cells, they play an important role in cell mediated immune response. The helper cells, they play an important role in both humoral and cell mediated immune response, and helper cells are capable of exhibiting two types of immune responses. They are T helper 1 response and T helper 2 response. This is T helper cell and this is T cytotoxic cell. Then receptors of T cells. The T cells also have some specific receptors on their surface. The most important receptor is T cell receptor. It is present on the surface of both T helper and cytotoxic cells, and it is occurring as a complex along with CD3, that is T cell receptor CD3 complex. T helper cell have co-receptors, that is CD4 on their surface in addition to T cell receptor CD3 complex. Similarly, T cytotoxic cells have co-receptor CD8. CD28 receptor interact with the molecules of B7 family of these B cells, and CD45 it is present on the surface of T cells and is involved in signal transduction. Then coming to natural killer cells, these are large granular lymphocytes. They are called null cells because they do not possess any of the receptors of the B cells and T cells. They have CD16 receptor on their surface and they attach to the Fc portion of immunoglobulin, mainly IgG, and they can kill tumor cells and virus infected cells. They can destroy the target cells by a process called ADCC, that is antibody dependent cell mediated cytotoxicity. Then coming to mononuclear phagocytes, these contain monocytes in blood and macrophages in tissues. That is, the circulating monocytes they differentiate into macrophages. Here, the monocytes they enlarges 5 to 10 fold and the organellar complexity increases and the phagocytic ability of the monocytes get increased and they start producing high levels of hydrolytic enzymes and thus they get converted to macrophages. Then granulocytic cells are the another category. These include neutrophils, eosinophils and basophils. Here we can see neutrophils they are multi-lobed and eosinophils they are bilobed and basophils are lobed, and the percentage neutrophil they constitute the largest percentage in the blood, that is 50 to 70%, and eosinophils 1 to 3% and basophils they are less than 1%. Coming to the staining character, neutrophils they can be stained with both acidic and basic dyes, eosinophils can be stained only with acidic dyes and basophils with basic dyes. The cytoplasmic granules, we can see primary and secondary granules in neutrophils and eosinophilic granules in eosinophils and basophilic granules in basophils. Then phagocytic ability, the neutrophils they are exhibiting the highest phagocytic ability and eosinophils are also phagocytic, but basophils they are non-phagocytic. Then other substances involved or present in the cytoplasm are hydrolytic enzymes, and that is neutrophils and eosinophils they carry large number of hydrolytic enzymes, but basophils they are mainly having heparin, histamine, serotonin, etc. The functions of neutrophils they play an important role in inflammation and they are capable of phagocytosis. Eosinophils they are playing an important role in allergy. Basophils are also doing the same function of eosinophils. Then these are the pictures of eosinophils, neutrophils and basophil. Here coming to next cell, that is the mast cell. This is seen in skin, connective tissues, mucosal epithelial cells of various tracts, that is our body tracts is covered with mucous membrane and these epithelial cells they all possess mast cells. They have large number of cytoplasmic granules and these granules mainly contain the substances such as histamine, serotonin and some other vasoactive amines. And the mast cells are also playing an important role in allergy and they are having specific receptors for the Fc portion of IgE antibody. Next is dendritic cells. Dendritic cells have long membrane-like extensions. They are resembling the dendrites of neurons. So they are called dendritic cells. We can see 4 types of dendritic cells. They are Langerhans cells, interstitial cells, interdigitating cells and circulating dendritic cells. Next is platelets. They are non-nucleated cells of the blood. They are derived from megakaryocytes. They have class 1 MHC molecules. They have receptors for IgG antibody. They are mainly involved in blood clotting and inflammation. They also carry granules like histamine and serotonin. So they are also playing an important role in allergy. That's all about the cells of immune system. Hope you will understand this lecture. Thank you.